so I think we should just continue.
Okay, as I was saying before I was so rudely interrupted, we've gone through some of the mandatory checklist of egregious errors that get into Cell Nature and equivalent papers. And now there are advisories, and the advisories are to use physiologically relevant conditions, as I said just before we were cut off, which is one of the things I was trying to do, modeling brown fat thermogenesis with isolated mitochondria, to ask the question, what occurs in the real cell and the real animal, and can we, as close as possible, mimic that response with isolated mitochondria, which is what we tried to do, I think, with fairly good effect. So these are not terrible errors. These are errors. These are bright scientists with successful labs who forget the physiological concepts, the physiological limitation constraints. There's one paper by our good friend Richard Yule on PINK1, and essentially the argument is that PINK1, which is involved in many hypotheses of Parkinson's disorder, that it is labile in response to changes in mitochondrial membrane potential. Mitochondrial membrane potential regulates PINK1. And so what some groups, for example, Ori and Shiri Hai, do is look for subtle millivolt variations in individual mitochondria and see what response that is. But the approach here is to add a bucket of CCCP, which is virtually the same as FCCP, for 3 hours, and then finding, not surprising, there's no mitochondrial membrane potential, but also that there has been a dramatic redistribution of Parkin to the mitochondria. But the problem is, what physiological condition corresponds to 3 hours of a high concentration of protonophore other than death? So it's a great pity, and that such resources go into experiments not thinking about what are the subtle physiological conditions under which this might be relevant. That I'm going to shift over. It's the whole question about that people have used the wrong conditions to stimulate insulin secretion for the last 30 years, which has led them to a lot of wasted effort and a lot of wasted hypotheses. Coming back to UCP1, but what I've been doing in my hibernation or my Easterbation here in the summer is preparing a UCP1 review of what's been happening in recent years. And you come across some wonderful experiments and some less wonderful experiments. So what we know from our studies is that you need roughly 100 nanomolar free fatty acid to activate UCP1, and it's sitting in a cytosol of about 5 millimolar ATP. So these are the physiological constraints for looking at UCP1 function. Now, I mentioned Martin Klingenberg, and Martin Klingenberg has made an enormous contribution to mitochondrial bioenergetics, particularly with the adenine nucleotide translocator. He's also interested in UCP1. He has purified it and reconstituted it and shown that it can be activated by fatty acids. But unfortunately, his fatty acid concentrations for activation are 1,000 times in excess of the physiological. And again, one thinks, how does one interpret this type of experiment? Is it that there is a 1,000-fold sensitizing factor which he is missing, or is he looking at something of no physiological relevance? These are some beautiful experiments that are published by the Kirchhoff group in San Francisco, where they have amazingly succeeded in patch clamping individual mitochondria, actually swollen mitoplasts, and directly measuring the proton conductance of these mitoplasts. The problem is that their sensitivity is such that they are focusing on this wide open conductance in the presence of fatty acid and absence of nucleotide, which is a conductance which is so high I can't measure it by my techniques. Whereas physiologically, the physiological range of UCP1 activation is from here to here. And it's unfortunate if one spends so much effort on a non-physiological condition, which for an otherwise excellent paper. ATP levels, this is a pet hate of both Martin and I. If you measure changes in just ATP, they're almost certainly detecting changes in pool size. So here's another of our famous Sirtuin papers here, where measuring the ATP level in skeletal muscle in mitochondria as a function of age, and claiming that the ATP levels drop by 80% between 6 months and 30 months, and saying this is a decline in oxidative phosphorylation efficiency with age. In fact, what it is, for whatever reason, is that the pool adenonucleotide pool size at 30 months is down by about 80% compared with that at 6 months, for whatever reason, if they are measuring it correctly. If the ATP level was down and replaced by ADP, this would be death. So don't trust pure ATP levels. Be a bit more trusting if you have ATP ratios. And I think, yeah, I will do this. This is a little bit unfortunate. It's something came up recently, and both Martin and I are interested in novel uncoupling proteins and whether they actually uncouple. And with the idea of going back to read the literature in detail, I went back to each of the initial reports for UCP2 and UCP3 between 1997 and 1999 at the very outset. Everyone was transfecting the novel uncoupling protein into yeast mitochondria and then using flow cytometry to monitor potentials. And they were using this diox-6-3. And at the time, it was considered that diox-6-3 measured mitochondrial membrane potential. But a year after this paper, a very nice paper by Hauger Rothenberg appeared, which said that if you're going to use diox-6-3, you've got to work at less than 1 nanomolar, whereas the experiments were working at anywhere 100 nanomolar, 17 nanomolar, 25, and 25 nanomolar. So they were in fact detecting a drop in plasma membrane potential in these original experiments, which was rather unfortunate. So there's our checklist, which is split between the mandatory and the advisory. And the advisory stops you wasting time, and the mandatory is absolutely essential. And the take-home message is you've got to take the time to read the methods section extremely carefully. It's not just a supplement afterthought that you've got to get online that you cannot find, as you know, it's not part of the main paper. It's the most important part of the paper. It's the evidence for accepting somebody's statement is based on the methods. Are the methods rigorous and correct? And in so many high-profile papers involving bioenergetics, I'm afraid they're not. And so the final conclusion is that particularly a high-profile paper is guilty and proved innocent, because science can be so misled by a paper, high-profile paper, being accepted as absolutely proven, where in fact the paper is flawed and the conclusions are flawed. So that's the end of my rant, and I will end the show and stop sharing, and we'll go back to everybody together. So we've returned and got our internets working again. And Martin, are you going to lead the general discussion? How should we do this?
Yeah, so this part is deliberately left unstructured so that any particular themes that you guys are interested in that you don't think we've covered, or we just briefly covered it and you'd like to hear more about it, or you have some question based perhaps on the project you're doing, but which would be of general relevance to everybody to hear the discussion of, this is the time to bring those things up. And often if there's a lot of seahorse users in the audience, we could discuss things that are particularly relevant to the use of the seahorse, like when should you normalize, or what does the various manipulations tell you about what's going on. Or we can widen that to questions of potentials and how you measure them, or bioenergetics more generally, or expand on points like pancashes. What's wrong with ATP? I didn't get what was wrong with it. So I'm kind of going to throw it open to you, and we can take this for as long or as short as it takes. We're making ourselves available for what, another hour, something of that sort, if you want it. But if no one wants to discuss, then we can wrap up fairly quickly. So really, it's up to you guys to run with it.
I have.
Oh yeah, go ahead.
Okay, Kevin first.
I wanted to ask about comparing and contrasting mitochondrial membrane potential versus mitochondrial calcium content. It seems that those dyes are actually somewhat interdependent and can overlap in terms of their effects. So TMRE and TMRM can be sensitive to the concentration and localization within the mitochondria of calcium.
Is that so? I'm not aware of that, actually. I mean, I've used the two indicators together because I've been very interested in mitochondrial calcium transport. But you've got evidence that is this optical overlap, or is it that calcium is affecting the fluorescence of the TMRM? What is the problem?
I think it's that additions of calcium affect the fluorescence of TMRM. And that absolutely could be a real effect on membrane potential, but I'm not sure that I have personally deconvolved that, but I've seen other people's evidence that they're concerned.
Okay, I mean, if you have a mitochondrial membrane potential, give the mitochondria calcium, the mitochondria will depolarize during the uptake of the calcium. And so that is a genuine effect, and you can actually see that with isotopes in the early days, excuse me, rather than with fluorescent probes. So, and certainly an effect you would expect to see would be a depolarization, for example, as calcium is taken up. Also, calcium has a very profound effect on the distribution between membrane potential and pH gradients, that if you accumulate calcium in the absence of a permeant anion, such as phosphate, then the delta pH increases and the membrane potential decreases. So there are lots of genuine effects there, and it'd be interesting. I mean, if calcium is affecting TMRM fluorescence per se, you should be able to see this in a fluorescent, in a fluorimeter, for example. I mean, the thing is that the free calcium that you see inside a mitochondria in the presence of phosphate is never more than about 1 or 2 micromolar. So you have to have that factor. I don't know, has anybody else come across this problem? It's not something I have. Any? Okay.
Maybe I could add that there are 2 more factors here that the mitochondria may swell. That can cause changes in redistribution. So if there's a greater mitochondrial volume, then mitochondria can take up more TMRM at the same potential, so that will cause an increase in the fluorescence. Other possibility, if you are in quench mode or you are close to the boundary between quench mode and non-quench mode, then the quench limit may change. And then what I'm wondering, that if you have a such high calcium uptake, that calcium phosphate precipitation happening in the mitochondria, that may have optical, like I think an absorbance to that. And I wonder if that can happen in a microscopy setting when you have very short light path to the light getting out of the mitochondria. So I don't know if that can interfere or not. I would assume it can't.
Right. I think what I've observed has most to do with alterations in mitochondrial structure and swelling in response to physiologically relevant calcium influx differences. And that's, I guess, what I've been most concerned about.
Okay, I mean, there's a basic rule which I tried to sort of talk about with regards to that apoptosis experiment. If your fluorescence probes do not make sense, it is probably that they're showing an artifact rather than you have to re-evaluate the laws of thermodynamics. So in other words, as everybody who uses these fluorescent probes know, they are tricky to use, basically, and there are all types of artifacts of different fluorescent enhancement and corrections for binding and so on. So, yeah.
My response to that would be, if you know that you've got volume changes, or at least physical shape changes, that's the most likely cause of the problem. Because if you have a redistributing probe, obviously the volume it's going into affects the total amount that goes in. If your signal is responsive to total amount in the mitochondria, it's going to change as the volume changes.
Yeah, I tried to use MitoTracker similar to the JC9 or JC1 type normalization. And basically it shows that ratiometrically there is an increase as well as the increased calcium. So I'm trying to get the pH measurements with the ciphers and the inner membrane and matrix to validate that it has a membrane potential as well as these changes. So.
Right.
Yeah, so that if you use a mitochondrial accumulating probe like MitoTracker, then I think that is an appropriate way to try and correct for volume changes, because that one would be subject to the same volume changes.
Do we know, does MitoTracker have the same quenching issues as the other ones would have?
I don't know. I've never used MitoTracker. Who's used
MitoTrackers are always in quench mode, I would say. So the problem with MitoTrackers that they are, all of them are potential sensitive at varying extents. So they will fluoresce in the mitochondrial membrane environment. Also, some of them may covalently bind, and all of them has membrane potential dependence. So it's very difficult to interpret a MitoTracker signal other than if you make proper thresholding and using it for geometric size determinations, like calculating mitochondria to cell volume fractions using MitoTracker statement.
Yeah. Another issue I've encountered is it depends on the cell line. So if you're using cancer cell lines, I mean, there's already low oxidative phosphorylation going on. Their membrane potential is already low. So loading with MitoTracker is sometimes difficult. Like I use SHSYs, which are neuroblastoma cell lines. MitoTracker green seems to be okay, but when I try to load them with TMRM or MitoTracker orange or red, it's like, it's hard. And when you increase the concentration, sometimes you might see it in the nucleus. I don't know.
Which cells were these, sorry?
These are SHSYs. They are neuroblastoma cell lines.
Do they have a multi-drug resistant transporter? Because this is the killer for so many cell lines. If they do, then it's not valid to use TMRM because the cell is trying to pump it out as fast as it's trying to equilibrate.
So another question is for everybody is like, now there's new palette of genetically encoded sensors which can be targeted to different organelles. Like, so I've used like GCAMP, which was targeted to mitochondria, and it seems like pretty good. And it's easy to focus the cells, do imaging-based analysis. I don't know what everybody thinks instead of using dyes.
I've never used it. Akosh, comments?
So yeah, measuring calcium, that's great. So as measuring pH, as ciphers were just mentioned. There is no genetic probe for membrane potential. I think there were attempts using the potassium, some parts of the alpha helical part of the potassium channel. I'm not aware anything that works in practice. And if it does, then maybe it may work better on the plasma membrane. I think that's a critical problem with the mitochondrial inner membrane, that if you were trying to insert there any kind of sensor that needs to be a transmembrane protein, that could have a high chance of messing up things there because there's such a high protein concentration in the membrane. So if one doesn't do it absolutely right, then you may end up with an uncoupling protein instead of a mitochondrial membrane potential sensing protein.
So at one point about calcium, I started working on mitochondrial calcium in the days when one used isotopes, one used calcium-45. And calcium-45, of course, measures the total amount of calcium inside a mitochondria, whereas calcium indicators measure the free calcium. And the free calcium can be only 1 or 2 parts per million of the total calcium. So there's this tendency in the literature to forget that one's measuring a very small proportion of the total calcium when one is measuring the free calcium with a calcium indicator. One has to bear this in mind. So you can load up mitochondria with almost 1 molar calcium before the cytosolic calcium rises above a couple of micromolar and you have the permeability transition.
On the same lines, people have used mitotimer as a way to gauge, I guess, the maturity of the mitochondria and like how that gets oxidized and the new mitochondria are produced. And this is, I guess, the probes available for all different species.
Anybody have experience?
I have a few people in my lab who work with mitotimer, mitoQC, or mitogcamp, I think, to varying extents. And I think a lot of them work, but if you make sure that that protein is not overexpressed to the point where it's aggregating, you also need to make sure it's localizing to the right place, because sometimes even if you have a mitochondrial localization sequence, you'll see very significant protein accumulation in the cytosol. But I think these probes are wonderful. I love the idea of being able to track how long the mitochondrion is alive, or whether it's being catabolized or not. I think they're super cool.
So another matter of concern is like how much you're expressing. So these calcium sensor proteins are itself buffers, like they have the EFN. So if you're expressing too much, they might themselves take up the calcium. So they might be like act as a calcium buffers.
There's Robert Campbell developed a number of ratiometric calcium indicators that are blue-green, or I think there's a green-red one too. They're called like Gen Gecko for genetically encoded, and then GECO for, yeah.
So when multiple constructs are available, then you are probably looking for the ones that has, if you talk about calcium measurements, then you are looking for one that has the lowest affinity, highest dissociation constant to calcium, because you will expect pretty high, above micromolar, or even higher calcium transients in the mitochondria. Maybe other interesting part that constructs that have tandem targeting sequence, like the 2MT, whatever we use the D4 CPV, a circularly permuted Venus CFP yellow chameleon version, and then the D4 that stands for a mutation in the calmodulin part that was providing a low enough affinity that seemed to work well in cortical neurons, for example. So I would just reverberate Daniel's and Manish's notes on the expression levels. So we've seen with mitochondrial targeted GFP that if you dose the cells with lipofectamine and something, you have a strong expression. You look the cell culture next day and you have green cells, and then you put TMRA on that, you will see that all those cells have barely any mitochondrial membrane potential. So you need to keep it in mind that simply overloading mitochondrial transport, protein import mechanism can depolarize mitochondrial membrane potential. So if you do such experiment that you have a culture and then you want to measure calcium or pH or use mitotimer, then you transfect the cells and wait a couple of days that the strongest expressors will just die off, and then you need to work with the mediocre expressor cells, not with the highest expressors. So in neurons, I like to do experiments only a week after life of action.
And on these lines, I just follow on from Pankash's comment that you need to worry about what your probe is measuring. So there are often things that are sold as redox probes, as if there's only one thing to be measured. And if you believe that and then you use it, you could be misled about the interpretation. So it's responding to something, but some of them are responding to thiol group redox state, others of them are responding to H2O2. And if you don't kind of bear in mind what the probe is designed to measure and try and use it to measure ROS, then you can get very confused because the ROS may be different from the thing you're measuring. The thiol group redox state and ROS are not the same thing. If you think they are, it's a problem.
Yeah, I mean, I agree, but just I think a lot of the probes, I guess the chemical probes are great for in vitro systems, but for more like David was talking about, more physiological measures and in vivo measures, the genetically encoded, I mean, I think a lot more work needs to be done on the genetically encoded sensors, but I feel that has a lot of potential as we understand these parameters, as we understand how proteins would respond to these changes, they could be quite useful. I mean, that could be the new generation of probes, like, perhaps.
Absolutely. I mean, the big advantage of the genetic probes is you can specifically target them to all sorts of different subdomains within the system in a way that an added probe you can't necessarily control. An example would be the MAMs. You can target things only to the mitochondrial associated membranes at the ER and interrogate that part of the system. And that's really hard to do with an added fluorescent small molecule probe. So yes, they have a great set of advantages. Chin is asking, does anyone have experience with genetically coded ATP sensors?
So yes, we do have. And that I just wanted to mention that, so I was saying a minute ago that yeah, you cannot measure mitochondrial membrane potential with genetically expressed probes, but you can measure ATP levels or ATP-ADP ratio, and then you can take these two as a surrogate of each other, mitochondrial ATP-ADP ratio or proton motive force.
So we are having a debate here in Sweden about whether Percival is actually measuring ATP-ADP ratio or simply ATP.
I mean, maybe if one is in the, so again, it depends on the experimental design, like the utility of the, maybe analogously to the utility of the quench mode TMRM, that if you are looking at acute changes, then you are not going to have pool size changes interfering because the pool size will change slower. So the mitochondrial targeted A-team, we use that in publication, for example, that you can get reasonable responses to varying substrate levels and inhibiting ATP synthesis. It responds appropriately. So along this line, interesting to know that you take in consideration the electrogenic nature of the ANT. The ATP-ADP ratio is much lower in the mitochondrial matrix than in the cytosol. And most of the ATP sensors that are genetically expressible ATP sensors have the caveat that they are too sensitive. They are sensitive to small ATP levels or too small ATP ratios, and they get saturated in physiological conditions. But if you use a mitochondrially targeted version, then it may just work all right because of the lower ATP-ADP ratio in the mitochondria. So in that way, in our hand, the lowest affinity A-team that didn't behave in the cytosol, but we could use it very well in the one that is targeted into the mitochondrial matrix.
I have two really easy questions and then one that's probably going to take a little longer to answer. One, are any of you or a subset of you that are teaching this course available afterwards for us to email questions if we think of something or want to talk about bioenergetics?
Martin is much better at answering emails than I am, but you can always try.
I'm very good at answering short emails. So if you send me one with 84 nested questions, maybe a month before you hear back. If you send me a short one with a one-line question, you'll probably get back an answer in an hour or two.
Great.
So send them to Martin rather than me.
No, send them to David as well.
I'll send to you both. And if, David doesn't answer, that's fine. Question 2 would be, are these lectures going to be digitally available since they're all being recorded, or is the recording for something on your end?
Vicky.
Well, that's a policy issue. So we have recorded them. We're not very experienced at it, so we're going to need to edit out a lot of the crap. The plan is to make them available, certainly to you guys. And we're trying to decide about what we do in the wider sense of, do we put them on the Buck website, or do we sell them to people, or what do we do? We haven't quite made that decision yet. But the plan is, that's why we're recording it. The plan is to make them available in some form.
But we're encouraged if you think they might be worth recording and replaying rather than just being consigned to history.
That's what we do now. We replay and replay.
Yep.
I have a question. We discussed a lot of isolation of mitochondria and looking at all this, but what do you do? Let's say you have a mouse which has an altered metabolic rate, let's say half the metabolic rate, right? Well, how would you take, and I kind of thinking of the questions that Martin, how he laid out his presentation in the beginning is very interesting, because they're, at what level do you want to ask the question, but how do you go from that point to sort of trying to explain how does your genetic manipulation or your compound have such a significant effect on metabolic rate of the whole animal?
What's the main determinant of
oxygen consumption.
Rate?
Sorry,
Yeah, but
I should use oxygen consumption per body.
Should one look at skeletal muscle? What tissue should one look at? Yeah.
So I don't know. I'm asking that. So
yeah.
Do you then dissect out different tissues and figure how would you go about from there to taking it all the way to understanding the molecular reasons behind what?
How far can you get with non-invasive investigations of blood flow and ATP levels, creatine phosphate, before you actually start taking the mouse apart? How much how far can one localize in the intact animal?
You mean localize which tissue it's coming from? Is that what you mean?
Yeah,
actually, the physical activity rate, let's say, is not different. So it's not at that level. So it's not the skeletal muscle, I would perhaps argue.
So the starting point for the way I would think about it is, you have to think about what determines metabolic rate. And in general, in a well-designed organism, it's not the mitochondria. When you don't set the system up so that your supply of energy is what limits you. You make sure you have sufficient supply of energy, and then you are limited by what you want to do with it. So metabolic rate is almost always demand driven, not supply driven. And obviously in the limit, if you starve something to death, it becomes supply driven. But biology takes great care not to get into that situation by having a whole range of mechanisms that stop you starving to death. So if you see a whole animal metabolic rate change, looking at mitochondrial function is not the place I would look first. I would ask, have they changed their blood pumping or their lung capacity, or have they changed their protein synthesis rates, or are they doing more maintenance or less maintenance than they were before, and so they're using a lot of energy there. But then, historically, people have tried to dissect basal metabolic rate and find out what it is. And one way to do that is to pull out different organs and to ask, particularly in a comparative sense, whether or not you see the same trends in the whole organ that you do in the whole animal. So you can look at the hearts from a range of animals with different metabolic rates, and what you find is that they tend to scale. So the hearts in animals with a high metabolic rate are also more energy intensive. And then when you try and work out the reason for that, it's because they have more of everything. They have more myosin ATPase, they have a more extensive sarcoplasmic reticulum, they have more mitochondria, and often those mitochondria have more cytochromes in them. So the whole system has been re-engineered to have a higher level of energy flow, because you need that in a higher metabolic rate animal, like a bird or a smaller animal compared to a large one. So it's kind of a long way from bioenergetics. That's not going to drive it unless you're really sick. And obviously in sickness, you can put some of the control onto the mitochondria. So human metabolic mitochondrial diseases would be an example of that, where if in fact your cytochrome B doesn't work very well, that's going to give you a metabolic problem, particularly under demand. So you have exercise intolerance often in metabolic diseases, even when they're mild. And obviously if they're non-mild, then you get neurological and developmental problems because you just don't have the energy supply to do the things you want to do.
But I think the inverse is also valuable. So if you have some sort of disease state or some sort of change in metabolic rate with a manipulation, and there's a hypothesis that it's because of mitochondrial dysfunction, whatever that means, you isolate mitochondria from the tissue that you think is relevant and you find no difference between the activities of the two samples. That tells you that the mitochondria are just as functional in the one as in the other. And then it tells you that the root of your problem is not mitochondrial activity, or at least not as you have measured it.
I have a question about the labelability of metabolites, because I like to work in an in vivo context. And what I typically do is I infuse 13C labeled metabolic tracers.
Yep. And
look at the flow of those tracers. And I know that when I dissect that tissue of interest, for me, the retina from that, there are going to be rapid changes in metabolite concentration. And I don't have like a microwave that can immediately, destroy all of the enzyme activities. Yeah. How should I proceed with this kind of work?
What do you want to measure? What metabolites?
We do metabolomics, and so typically it's a lot of TCA and glycolytic metabolites.
And you're starting from the intact animal, and you have to dissect out the retina? Right?
Which honestly, for the retina, it's relatively easy, but for other tissues, it's not so easy.
You're using stable isotope tracers as well, which is a much better measure than what was shown earlier, which was just the total metabolite. So you actually see conversion and incorporation.
Yeah, but all the same, I mean, I don't know to what extent that's going to change instantaneously after I sacrifice the animal and before I dissect that tissue.
So your question is an experimental one of how do you get a sample which represents what was going on in vivo?
Pretty much, yeah. Any ideas from anyone really about how to best approximate in vivo with these methods that I currently use?
You've got to decrease time as far as possible, and that's limited if you've got to dissect out the retina before you do anything, isn't it? How long does that take?
1.5 to 2 minutes.
Yeah.
And historically, the way that people approached this problem back in the 60s and 70s in other tissues, particularly liver, was to progressively decrease the time between death and when they had their sample and find out if they were still getting differences. And they decided they'd reached the answer when it didn't matter if you halved the time or took the time down even further. And with a metabolically active tissue like liver, the way to do it was freeze trap. So you would open up the animal, hope that nothing much was happening, and then you'd go smash with some aluminium tongs that had been stuck in liquid nitrogen so that everything just froze like that. And when you do that, you can get from in vivo to sample within a second or two, within the time it takes to freeze a sample. And what they found is you really have to do it very fast, like within a few seconds, when you're looking at things like TCA cycle intermediates or ATP and ADP nucleotide pools that are turning over really fast. Of course, that statement is not true for things that turn over slowly. So levels of RNA, you can put it on the bench and go and have a cup of coffee and come back, and mostly they won't change very much. So a lot of the molecular biologists who come into the field come in from proteomics and nucleic acid chemistry, where the time is really not so important and they've kind of become sloppy. So in your case, what ideally you would do, and technically this might be difficult, would be to freeze clamp eyes and then dissect out the retina and see whether you get the same answer as you got in your a slower dissection procedure. And if you did, then you're fine.
I've also seen people use a control of a pulse chase in between the harvest time period. So if you were to add in your labeled glucose at the time of sacrifice, or once you have the retinal sample in your tube, to see what the metabolic flux differences are after harvest, and then subtract those from the thing you think is the in vivo flux.
Okay, good idea.
I'm actually not sure I understand that point.
So if you harvest the retina and then incubate it in an equivalent concentration of your labeled glucose and see that there are still appreciable changes in the labeled metabolome, you can assume that that's an artifact beyond the in vivo metabolism that was observed. And you can subtract those rate differences from your actual sample to say that those might actually just be an artifact post-handling.
So that's a good control, but it's a control for continuing metabolism once you've got the stuff in the tube.
Yep.
It doesn't control for what you would like to do, which is metabolism between the time it was in vivo and when you get it in the tube. Although in principle, you can extend it to that. I mean, you could inject C13 standard into the eye and then see whether or not you find it all at the end. And if you don't, then clearly you weren't capturing everything.
I have a question. If we're on to the next one.
Yeah, go ahead, Richard.
So when I just started playing for the first time with a Clark probe, and so I was checking the voltage of a solution before, and just by inserting the probe in the solution, it changes the voltage of the solution. So I'm wondering what effect that has on the experiment. Do you follow what I'm saying?
I'm not quite sure what you mean by the voltage of the solution. Can you?
So I measured a solution before, just using a voltmeter, and then I measured it afterwards. And once I with the probe out and with the probe in, and the voltage drops about, I think, 90%. And so I'm wondering, so just by inserting a probe, you're affecting your readings, but I'm just wondering how that would.
This is a Clark type oxygen electrode you're referring to?
Yeah.
That should be totally electrically isolated by the membrane
No.
over the end.
So when you ground something, so the probe is grounded, and when you ground something, you will change the voltage of what you're grounding, because it's the product is isolated. The solution is isolated until you introduce the probe, and then the probe itself has some grounding effect on the solution.
Yeah, I think that is
hard to explain. What?
I think that's what's going on. So the two things are quite separate, I would say. That the Clark electrode, as David said, is electrically isolated. It doesn't care what the voltage of the system you're dumping it into. But it is itself a way to ground that solution. So historically, when we were doing experiments with that sort of system, we often used to dip pieces of wire into solutions and then connect them to metal rods beaten into the earth outside the lab to really ground everything. But the reason for that was to remove transients. So if you're trying to measure things with rapid time courses and the ground is changing because your solution is partially isolated, then it's very sensitive to people walking past, or if you put a metal pipette tip or a syringe into the solution, then you become the grounding circuit yourself. And to get rid of all those artifacts, you can ground the solution. But it doesn't affect the signal that you get in terms of the rate from a Clark electrode. What it affects is transient spikes. And if you're looking at very rapid times, that can interfere, particularly if when you make your injection with a microsyringe, you also ground everything. So you get a big spike at time of addition. These days, we've switched to using fluorescent equivalents of the Clark electrode just because they're more stable and easier to work with. So I was a bit hesitant, but when we bought it, we discovered actually this is really good. So we now use that in preference because you don't have all the messy changing of membranes, and you don't have to worry about the current flows because there's no net oxygen consumption. So they tend to be a better way to do it.
You could put 24 of them together and make a seahorse.
If you don't have money.
Yeah.
Any other points?
Plenty of people seem to use their seahorses as respirometers and nothing more. So.
I have a question about determining the proportion of your cell that's occupied by the mitochondrion. It seems to be a coefficient that you use for several things.
Yeah,
and I was wondering if you've compared the technique that you had mentioned before, using the 14C sucrose and what was it, tritium or something like that.
Tritiated water,
if you made it, I'm sorry?
Tritiated water and 14C sucrose.
Yeah, I wonder if have you ever compared that with something like electron microscopy, if that gives an accurate report of how much cell volume is occupied by matrix?
No, but Akos has done something rather similar to estimate mitochondrial volume percent. Do you want to talk about it?
So we use confocal microscopy for that, and here is MitoTracker green, is MitoTracker red in this case coming back into view. So what we do is, it's not an isotopic method anymore, but we take high resolution single planes of a MitoTracker stained cell. We stain the whole cell with calcine AM so that we stain everywhere, nucleosol, mitochondria, cytosol. And then we use image binarization. So we determine that which pixels are belonging to mitochondria, which pixels belong to the cell. We use some basic serologic considerations, just as one would do it in electron microscopy. And we compare, so we calibrated this technology based on electron microscopy. So we took the same cells that was a insulinoma cell line. We put them into the electron microscope, and we imaged that with a confocal microscope and adjusted this algorithm in a way that it gives the same value. Now, we think that it works relatively broadly in terms of using different cell lines, as long as you are in between this 5 to 10% of mitochondria to cell volume fraction that you see in typical cells, let's say ranging from fibroblasts to primary beta cells, primary neurons. But when you go to the extreme, like cardiomyocytes that have larger mitochondria and a much higher mitochondria to cell volume fraction, it's about 30%, then such algorithms need to be retuned. Also, if you go to a different microscope, it may need some tuning. But when your question is, David?
So one point about the sucrose, the mitochondrial matrix volume, is that we occasionally make the mistake of thinking that it's a nice, pure, dilute solution. So a typical matrix volume of 1 microliter per mg of protein, roughly half of that protein is free matrix protein, which means basically you're looking at a matrix which is virtually 50% protein. It's a thick soup of concentrated protein, and yet we still believe we can apply Nernst equilibria to it after correcting for binding. So it's a very non-ideal solution that we tend to ignore when we're measuring our potentials. We do corrections, but do we do enough correction?
I think we don't ignore, and also like Martin's early experiments didn't ignore. So one can introduce one can talk about chemical activities. That's a generic term that can include this dilution of the aqueous space by protein. So the chemical activity of TMRM, when we calculate by our own means, is much lower in the matrix than in the cytosol, which can be partly due to binding to membranes. It can be partly due to this reduced aqueous space, so the large spaces taken up by proteins. So therefore, at least in our calculations, we define the mitochondria to cell volume fraction strictly as a geometric term. So that's a total ratio of total volumes. And then we use another factor, an activity coefficient ratio, that is possible to measure just based on taking fluorescence of mitochondria in a cell where you completely depolarize mitochondrial membrane potential. And that we believe at least that, and it seems so, that is useful for correcting for this matrix, high matrix protein content.
Yeah, I mean, you can do that adjustment in live cells, and so I'm much more trusting of that than I am of the EM data that I can get more easily. I guess I'm just concerned that there's a fixation artifact which affects mitochondrial size differently than it affects plasma membrane or cytosolic size, I guess.
Yeah, that can happen.
Yeah, actually the mountains that you often use for stabilizing samples will flatten cells. And so you should be really careful about doing that. Not to mention, if you're going to use a confocal, you might as well do the full stack so it's a real volume and not just a surface. Because
Go ahead.
So what I've found is that the way the mitochondrial network wraps, there's some very density differences perinuclear versus to the periphery of the cell borders. So if you flatten your cell, you're losing cytoplasmic volume and you're looking to actually consolidate some of the mitochondria that's really in the dense areas. So it's precarious depending on which mountains you use. I usually use the Pro-Long Golds, and those are actually notorious for flattening cells significantly more than glycerol or other stabilizers. So you might want to look into that.
Yeah, so yeah, you're absolutely right with this point. So therefore, we do it on live cells. So the mitotracker to calcium ratioing is strictly with live cells. What you observe, the effect of prolonged gold is because of the differing refraction index. So there's a refraction index mismatch between your immersion oil and your aqueous medium that distorts the Z scaling of your Z stack. And yeah, there are technical reasons here. So we don't make Z stacks because that would just burn up the cells. So we take, therefore I'm saying that this technology is like classical stereology, that we take single snapshot images at different Z distances from the cover slip, and one per cell or one per area. So we don't repeatedly image the same place in the culture, because we know that the second scan would be already different, and that would be a photodamaged cell whenever you want to take a high quality, high resolution image. But you had you were right on the point.
I see that Gulchin has a question in the chat window. Do you want to go ahead?
She can't use her volume for some technical reason.
I'll go ahead and read it. Could we also discuss seahorse data normalization protocols? I noticed the seahorse data used in problem sets were based on total protein amount. Yeah, so we can talk about normalization, and you're right that we have shown a lot of data normalized to total protein in the well. It's not really a question of the right or wrong way to normalize. It's the normalization that best fits the question that you are asking. So however you choose to normalize is an implicit part of your hypothesis. So if you're normalizing to total protein, you're saying that any rate changes that you want to detect are relevant on the level of the protein content of the well. You might normalize to cell number because the measurements that you're looking for are relevant per cell. So it really depends on what you're asking and what you're trying to measure.
So an example of that would be, imagine that your treatment, what it does is to proliferate the mitochondria. So you have twice as many mitochondria, but they're otherwise the same. If you normalize per cell, what you'll see is a doubling of all your rates. your mitochondrial determined maximum rates at least, and a change in the other rates, because you've got more mitochondria doing the same thing. And if you care about that, then you would normalize per cell, because you would see changes in rates. But if you know that's an inappropriate part of what you're looking for, because you're looking for a qualitative change in those mitochondria, then you want to normalize that away so it doesn't interfere. So instead, you would normalize to the amount of mitochondria in some way, like the citrate synthase as a surrogate of amount of mitochondria. And then all you would see would be changes which would qualitative changes in the mitochondrial ability to do something for mitochondria that are actually different. So both normalizing it per cell or per total milligram of protein or something, or normalizing it per citrate synthase are valid ways of doing it, as Shona said, but they give you a different perspective on your data. And that perspective has a hypothesis built into it, as she said. So you have to normalize appropriately for your question, and there isn't a single normalize like this answer. I think Seahorse at the moment are trying to sell you a kit which says normalize like this, but that avoids addressing the issue that no, it depends what your question is as to how you should normalize.
Can I actually show you guys some data that maps all the different ETC subunit changes, and then get your opinion on which protein should I theoretically normalize to in the future? Because this is a big problem that I've run into, is if I conditions that function for the way the electron transport chain alters, and I'm normalizing to total protein, that's not really a fair representation of respirometry, right?
It depends, right? Doesn't it depends on how representative the total protein is of the change in the complexes that you're trying to monitor?
I mean, I've reached the point where I kind of feel like maybe I should measure like cytochrome C, but these are the fold changes that I'm seeing proteomically between two conditions I'm interested in comparing. And there's a lot of changes that make it difficult for me to make a decision on which complex I should be looking at.
I mean, the other issue is that you don't necessarily know whether the abundance of particular subunits, how strongly those affect the flux through the system that you're measuring.
Right.
But in the end, it comes down to what question are you trying to approach here? And you can't approach the question of how to normalize until you've answered that question. So what do you want to know? Do you want to know if the mitochondria behave differently? Do you want to know if there's more limitation by complex 3 than there was before? And what do you want to know?
So right now I know that bulk respiration is suppressed, and I know that there are compositional changes in the electron transport chain, and based on abundance I can make some assumptions about where those changes are. But since there aren't really assays to test every single subunit, or that would just be more work than would make sense, I'm kind of at a loss for how to compare.
But just to check, your respiration limited is being limited by the electron transport chain. We're not looking at a limited respiration due to ATP demand or photon leak. You're focused on the electron transport chain.
Right. There's no difference in uncoupled or state 4. So.
Okay, so your question is, you see a change in maximum respiratory capacity, you see a bunch of changes in subunit composition, and you want to find a causal connection and say it's this one that's the cause of it, not that one?
Yeah, ultimately I would like to figure out where that is. But I was thinking about using different genetic tools to tune the relative abundance of each of these things, but I don't think that I'll be able to exogenously enforce these differences synthetically. So I'm not sure how to go about conceptualizing a normalization strategy to figure out where those differences are coming from.
You've got a couple of sites. You have a couple of sites you've got access to. There's cytochrome C reduction. There's the UQ reduction state. So you can detect in which region, which complex region, the control is altering in your condition. I mean, so is complex 1 going more reduced, or is complex 3 going more reduced, or you might be able to sort of localize in a bit, find out where the flux control is changing.
I mean, I think the automobile factory is a good analogy here. You have a factory that's empty, but it's full of machines. you have different numbers of different machines that make different bits, but you don't know how quickly cars are being made or how quickly cars can be made until everybody comes to work, you start making cars, there's a rate of car production, right? And so then you say, well, I want to know why that rate of car production exists. I'm going to take out this machine and I'm going to see what happens. And that may or may not explain the change that you see, and it may be distributed across the addition and subtraction of multiple different machines in different parts of the factory. So it might be folly to try to pin it on one. That would be like pinning it on a rate limiting step, which we discussed earlier doesn't necessarily exist.
Have you tried permeabilizing the cells and run like similar experiments that you just learned, the electron flux experiment on isolated mitochondria, but using permeabilized cell cultures?
Yeah, and different substrates. That's exactly where I was going with this. And it does look like there's a big block at complex 1, but that's kind of what I was getting at, to figure out how to basically infer where the biggest differences are. But since I'm seeing these compositional differences that go up and down throughout the ETC and then affect the total, affect possibly the super complex assembly, I'm realizing that these questions are very, very difficult. And I was kind of considering if there was something I could normalize to that would abolish the difference, then that might actually suggest to me that's where the effect is coming from.
Yeah, that would come back to what I was saying, the citrate synthase. So you could start from the hypothesis that one of the complex 1 subunits was completely determining this effect, and the rest of them are just noise around that. Yeah. And you could, by Western blot, normalize to that subunit, and if your rate changes or went away, you could infer that that's the rate limiting subunit, that's the only one that matters. But then, as you're recognizing, the world is unlikely to be that simple. So it's much more likely that you're going to get a mixed answer where it has some effect, but it doesn't make the effect go away completely. And then if you were to do the same analysis with some other subunit, it would also have some effect. In other words, the control is distributed. And your basic model here is one in which the control is not distributed. It's on a single step, and that, I think the world doesn't work like that. So you can't normalize your way out of a complex problem like that.
I have a question about how could you give us the latest in the developments of how seahorse has been used for organoids and tissues, and also like I'm thinking about C. elegans or parts of a fly and things like that. What is the latest in that?
Seahorse have stopped talking to me for the last 5 years, so I can't actually update it if Martin has anything more.
Same thing, that we can read the literature like you do, so it's just a matter of really anybody in the audience that's come across particular examples of things that they think are neat. But certainly, yes, you can use cells. I've come across papers where people have used tadpoles in the seahorse or embryos, and clearly things like islets. So you can look at respiration of that. I've come across organoid papers where people have made organoids and then shoved them into a seahorse and measured rates. So I think all of those things can be done. The normal problem is sticking the material down, which is what we do with the isolated mitochondria, is we centrifuge them to stick. Because otherwise, when you mix, they all get mixed up, and then next time the plunger comes down, there's less material in the well. So you want something that's either big enough so it always ends up in the bottom of the well, or you have to hold it down there somehow, either by gluing it to the base or spinning it down, or in the old eyelet plates that seahorse used to sell, you put a net over it and just hold it down at the bottom.
And the other, so one is the fixing, and the second part is as long as the tissue can, as long as all the inhibitors can get into the tissue, you should be fine, right? In principle, that's all you really need. Those two things, the thing has to stick and then has to be perfused.
Yeah, it has to be perfused. So obviously you have to worry about oxygen getting into the center, because if you have a bunch of 100 cells all stuck together in a sphere, the cell in the middle may not be getting the oxygen at all. It may be anaerobic. So at some point the thing falls down because you don't have a native circulation there of oxygen.
A second consideration on all of this is that the further you get away from the ideal and recommended conditions for the seahorse, which is 37 degrees and air saturation, the constants that are used to determine the oxygen consumption rate are less and less applicable. So people have tried to use the seahorse at varying temperatures from room temperature to 30 degrees to 37. And the constants underlying the calculation are optimized for 37 degrees, for example. And so the more you manipulate the system away from that set of parameters, the less well the rates actually fit what's going on inside the measurement well.
Yeah, in line with the use of C. elegans, first question was for me about the temperature, because seahorse works at 37. At that particular temperature, the C. elegans would be completely different. It is not an optimum temperature for C. elegans. And the second thing is that so far we have talked about an isolated mitochondria or a certain type of cells. But in case of C. elegans, it's going to be a multicellular system where cells are talking to each other within the system too. So how are we going to control all the parameters that we discussed like for last 2 days? And how can
if you stick a worm in the seahorse, it's basically like sticking a mouse in a metabolic cage. You get an input and output from the whole animal, but the animal itself is a black box. And what you're measuring is the sum or net of everything that comes out of the animal.
Right. So during data analysis, what are the things that I need to be very concerned about when I'm working with the C. elegans? So because that is the most important part, right?
To the question. One is temperature. So the seahorse, as you say, is designed to work at 37. If you turn off the temperature control, it will drop down to a few degrees above ambient and wherever you have your machine. And in principle, that can be in the cold room. So you could run it down to a fairly low temperature if you wanted to. The problems are all then technical. You get condensation problems because it's not designed to work there, and the electronics may not work properly. All of the correction algorithms are devised for 37, so you're kind of taking a big risk. You can get qualitative information, but you have to be very careful about quantitative information. And then the other aspect is the complexity of what you're putting in. So if you put a worm in and then add FCCP, it's not going to be like adding FCCP to a cell, because the worm is obviously well built to resist nasty chemicals in its environment. So you're just reliant on what it will be prepared to ingest, and then the ability of whatever it ingested to get to all the cells from the gut. And some things do get into worms and do distribute, and other things don't. So it's very empirical, and then they're having different effects in different organs. So it's really hard to do a clean bioenergetic interpretation in such a complicated system. So as Sherman said, you can get the in out, how much oxygen is the whole thing using, you can get that, but you just don't have the ability to make those precise changes that you do in the cell experiments or the isolated mitochondrial experiments. I guess, Pankaj, you have some experience of using worms in the seahorse, right? Or Gordon does, I suppose. You're muted.
Yeah, Gordon's lab does it, but I'm looking at the papers. I think you're right. I mean, oxygen consumption's about as far as people have gotten, really.
Mm.
And under different conditions, but the problem is the way the toxins go, yeah.
Mega,
we would love to know the spare respiratory capacity. So that's the question I have.
Yes.
Yes, worm. What is it? Yeah.
Put it on a treadmill and exercise it.
Uh-huh. Yeah, push it. Okay.
So, I mean, any system more complex than individual cells is going to, I don't know, it seems like it's difficult to use seahorse specifically, you know, in more complex systems generally, because you don't know which contributions which cells are making. Is it even worthwhile to pursue bioenergetics in a more complex system than a cell?
One of the more complicated is a pancreatic islet. And an islet is a collection of 100 cells of 4 or 5 different cell types. And the information one gets out of that is really limited because of the heterogeneity.
I think, I mean, overall, the question is just another level of abstraction from what was presented earlier about the pluses and minuses of isolated mitochondria and then the pluses and minuses of cells. When you extend that further out to the pluses and minuses of organoids or tissue slices or worms, you are creating what is a better physiological context with less experimental control each time or with each level.
Right.
And so as long as the benefit of the data outweighs the drawbacks of the experimental limitations, then the answer is yes, it's worth it. But as soon as that balance shifts towards it's not worth it because the experiment is too limited to be useful, then it stops being worth it.
The other way to expand that is to think about all of the muscle physiology labs around the world who take humans in, put them into calorimeters or into huge respirometers and measure their oxygen consumption. And what you're saying is that's a waste of time. And what they would say to us is what we're doing is a waste of time because we get rid of all the interesting sports physiology and we're just looking at stuff in a test tube. And what does that tell you about how fast an athlete can run? So it's all a matter of perspective as to whether you want a physiologically relevant answer or whether you want a mechanistically precise answer. And what we all want is both, but you can't have what you want. So compromises.
So I was, I mean, taking up on what David said. So, I mean, if you just take it from first principles, and if you're trying to understand the different mechanisms, but instead of using these compounds, if you could stress an organism in different ways. So we have that ability, which you can't do with a human, but like something like exercise or something push, or like even changing with temperature, how far can you push this system? I mean, we're talking about how to raise metabolic demand, right? And we could do that with temperature, for example. Would you learn something by changing all the oxygen, learning about just oxygen consumption by changing all those parameters? I mean, you could imagine coming up with things equivalent to the what you have with the pharmacological compound.
Sure, I think temperature is a difficult one because you change the supply and the demand simultaneously. So what you really need to do is train your worms to run on a treadmill, and that would be a better way of getting an increased ATP demand.
Worms can be done. Worms can go for exercise thing, so maybe we can try that.
Yeah, we can talk in a smaller session. Yeah.
We have a couple. Oh, sorry, Martin.
Yeah, you're going to do the same thing as me. So Mega asked the question, is there a good reference for troubleshooting a seahorse experiment? I've heard there's a lot of variability in the hands of new users. So the answer is yes. If you don't really know what you're doing, the seahorse is pretty adept at making you look foolish. And we went through some of the ways that you can get the wrong answer due to straying outside the parameters that seahorse recommend. In terms of the rates that you're measuring or things that change during a measurement, as in when the ADP runs out, for example. Probably the best way is to, I mean, this is like company propaganda, but is to get hold of the company rep for your area and get them to come talk to you and go through your experiment with you because they've had experience of all the different ways that users can get it wrong and may be able to help. And the same would be true for an Ouroboros. If you contact the Ouroboros company, they have a big database of users, and I'm sure they would help you to do it. You can try and read reviews and the various articles that are published, but troubleshooting, it's hard to do troubleshooting that way. It's much easier to ask a person.
Yeah, but there are a number of things I think that we've covered most of that are easy to do. And and they mostly have to do with signals that people don't always think of to look at, but you should. So you should always look at your acidification signal. You should always not only look at the rate, but look at the pH level to make sure that your medium is pH'd properly. So those are a lot of the common reasons that things go wrong. But then if you take it backwards to even just setting up the plate, there are a lot of technical reasons that you can inject material right through the port into the well. So all your material is dead by the time you put it in. So there's some troubleshooting that is setting up the plate, and there's some troubleshooting that's making sure your medium is what you think it is. And then there's making sure that all your signals line up in the way that they should, given what you think you've put into the plate. So a lot of that can happen without asking anybody, just looking at all the measurements that come out of the machine.
But please design your own experiments and don't buy very expensive kits which actually leave you with no flexibility. I mean, as we've tried to discuss the last 2 or 3 days, you should have enough knowledge to design precisely how much uncoupling you're going to add, how much membrane potential indicator for doing that type of experiment. Just buying a kit is simply a way of wasting money and restricting your flexibility. So we don't like kits.
I guess this is the way that a lot of users might think of a Seahorse as an easy bake oven, where you have to buy the easy bake oven cupcake mix to be able to use it. And we don't use it that way. We buy flour and sugar and eggs, and then we make our own cupcakes.
And
So
practice common sense. So I've seen several examples, a lot of it. So like people just grabbing the cartridge and putting out to the bench while pipetting just as it is naked with the probes on a bench. So think of contamination. Do not contaminate in any way your probes or your cells in terms of with the chemicals that you are using, or do not stab the addition ports while adding the loading them with addition. So these are all small nuances, I would say mechanical issues that you need to just keep in mind. And they are just basics. Just look what you are doing. Look at that your cartridge didn't dry out while hydrating and such. So with care, you can do very precise experiments, and with sloppiness, it's very easy to make unpredictable data.
And part of the reason we're saying all this is because a lot of the variability by new users are things like this. They're easy to avoid once you know what to avoid. And a lot of it's common sense, a lot of it's making sure that everything that all your material is what you think it is.
And just repeat that, look, and look. So before you load the cartridge, you have a glance on it. If all ports are loaded, you can hold it in front of a black screen or something, and then you will see if there is liquid in all ports. After the experiment, you look again. You look whether all ports have been injected, because it sometimes fails to do so. Then you look at the plate, are cells still there, or maybe they got washed off during handling. You can easily troubleshoot experiments just by having a strict eye on them.
Yep.
So there's a question from Kartik of normalization, so using nuclear stains or mitochondrial dyes. So I think, yeah, again, the use of whatever normalizing agent you choose depends on the question that's being asked. I don't know if there are additional artifacts that could arise from dye sensing that, yeah, doesn't reflect the material in the well. So that I don't have a lot of experience with.
It's like phenol red could interfere with the probe, therefore all the media are in phenol red free. But I think Martin, we had some other thing which was like very strongly colored and it was clearly interfering. I don't remember what was that. So if you put a colorful or fluorescent thing into the wells, then think about it.
You can do it after. You wouldn't want to do it before, but if you're doing like a live dead stain after, or a mito tracker stain, and then just scan your well,
or something like that.
I mean, the reason I'm asking is CS is already expensive, but now Agilent is collaborating with biotech and selling a combo with Citation 5, and several people are using it. And even like Orient Sharia in
better mousetrap, right? If it's
I mean, we do the same thing that we don't use a biotech, but we are using a Nikon microscope.
Yes.
We plate after readout, fix it, stain it, and count the cells. So yeah, that's a legit normalization. That's very good that they are selling it in a ready-made system. But you don't need to buy their biotech citation. You can use any other available imager to get that count, or you can measure protein.
But one thing I have noticed with the SciQuant data is that people don't tend to calibrate it to number of cells. They'll just put the fluorescence value on the y-axis. And that might be fine, but it might be misleading if there are different reasons for changing the fluorescence signal when you're comparing plate to plate or comparing between cell lines or samples.
But also we may need to take 2 steps back along this line, because the Seahorse is it's really great for working with baseline data. So if you can design your experiment in a way that you can make a legit baseline normalization, so all wells start in the same conditions, and then the additions are different, then you can achieve much greater accuracy than if your design is a pre-treatment and wells are different and then you are trying to normalize. The machine is a lot more sensitive and precise on baseline normalized data than well-to-well comparison, even if you include fancy normalization like citation 5 or protein measurements.
I have one more question regarding the ATP calculation. So you mentioned that it is better for one substrate at a time. I think you mentioned yesterday, like if we play with multiple substrates, it will be rather difficult because you may not know what % is being utilized, right?
Sure. And I guess I wouldn't say better or worse. I would say we have more confidence and less confidence.
Okay.
Because the assumption of exogenous use of a single substrate is more likely, and then we can assign all oxidation to that substrate. Whereas for mixed substrates, we don't necessarily understand how distribution of those two substrates occurs.
Okay, and even in that,
people haven't done this, so.
Yeah, even in that, in those calculations, at least for the oxpas, I just quickly read through it. There were like 2 shunts where the NADH can come inside the mitochondria, and it is assumed that 90% is through the glutamate.
Aspartate shuttle.
Yeah, malate aspartate.
Through the glycerol phosphate shuttle. Yeah, so to the extent that those aren't true, those P to O values will be in error. But you can work it through, and we have, and the error that occurs when you're wrong about that is still swamped by the measurement error of the machine. So if you're using, for example, brown fat, and you think that a much higher proportion of reducing equivalents come into the matrix through the glycerol phosphate shuttle, then you might decide to change that ratio and therefore change the P to O value that you plug into the spreadsheet. But any changes that you would make in your assumptions would be reflected in one of those ratios, either one of the ATP gearing ratios of ATP to lactate or one of the P to Os, or your assumption about H plus to O2, so the ratios of the measurements.
Thank you.
But all of the assumptions then, get applied to those ratios. So there's the stoichiometry of complete oxidation, which is also an assumption. If you assume it's completely oxidized, the stoichiometry is fixed. Everything else is dependent on the assumptions of no growth, complete oxidation, and the shuttles operating in the way that we predict that they are.
With regards to that, I'm curious if there's a way to sort of integrate metabolic flux information as a way to sort of get a bit more accurate, might not be the right word, but to gauge a little bit more into that. And I'm just curious if there are specific tissues or conditions that people know where that sort of goes out of whack, where you have metabolite levels and metabolite fluxes not really going hand in hand with bioenergetics, whether that is reverse electron transport or something like that.
Right. So for the first part, it would be ideal to assume less about the P to O ratios and either be able to measure them directly or have like carbon flux data that gives us a better sense of proportional use of different substrates. And then we could refine the values that we assign to the P to O. And the second part about tissues doing weird things or tissues that are known to be unique. Was that the question, Carl?
Yeah, and sort of, if there's sorry for the background, I don't know what that is. If there's sort of a good way to sort of gauge on the bioenergetic side and either from Seahorse, sort of put those two sides together to get a fuller picture.
So have we combined existing metabolic flux data with our assumptions to try to refine the calculations? Not as yet.
So I'm new to the side of bioenergetics, so I've been recently working more on the metabolic side. So I'm sure that the examples are out there. I'm sure there are tissues and cases where those don't go hand in hand. I was just curious if people had seen them or experienced them and sort of how they dealt with that.
And I think the general point here is that in any specific question that you're interested in, you probably wouldn't end your whole measurement with a seahorse run or a membrane potential run, because you have a wider question, and you would bring in other techniques and you'd try and build a picture where everything was coherent and you could correct one technique with what you know from another technique. So we're not saying that to do the cellular bioenergetics is the be all and end all, but it's an easy and quick way in, particularly if you have a seahorse available to you and you don't have mass spec flux facilities available to you. If you have those things, then maybe that's where you would go to start with. You wouldn't go through the indirect methods of using the seahorse. So it's condition dependent, and we never think you're going to stop with just one measurement when you have an important question that you're trying to answer.
But it's also true that a lot of metabolic flux analysis traces carbon, which ATP turnover is invisible to. So yeah, as Martin said, you can find another way of measuring it. Oh, sorry, Daniel?
Oh, I was just saying you can use isotopically labeled oxygen, I think, or yeah, oxygen, and that should be able to trace ATP flux if you're careful with it.
Yes, I mean, there are technical issues there which to do with exchange reactions versus net fluxes, but yes, all of these things can be resolved, but you have to dedicate your whole life to it to do it well and to really get the information you want. Whereas a seahorse, you just dedicate a week. Okay, so we're probably getting close to wrapping up. Are there any other issues that people really feel they want us to cover, or is it now time to draw a graceful end to our proceedings?
Well, for me, it's 3 minutes to midnight, so I should be leaving very soon.
Martin, I have a question about your Amplex Red measurements. There are mitochondrially targeted hydrogen peroxide sensors now available. One's called Mito PY1. Have you done the assessment of contribution to hydrogen peroxide production, comparing the Amplex Red to, say, a Mito Matrix sensor, and seen the same proportion or different proportions?
No, we haven't tried that. Remember that what the Amplex Red measures is a rate, and what most of the probes measure is a level. So to get rate information using a probe of level, it can be done, but you have to do sequential measurements, and you have to know that other things are not going on and interfering in time. So where we are at the moment is what we're doing works and is internally consistent. We should try and use some of these probes to cross-check that we get similar results with them. But our experience of the probes generally is that when we've done that, it's been a rabbit hole of lots of pain and unhappiness, and we don't get anywhere. So I'm kind of slow now to go down that path, but it is one we should do, and eventually there will be probes that will be good. So at that point, we would switch to using them. Okay, well, thank you very much, everybody, for sticking with us. There's still 38 participants, and I take that as a compliment to us. I hope you've enjoyed it, that it's been a worthwhile way to spend 3 hours sitting in your chair in front of your computer screen, 3 days. I'd like to thank David for, as ever, wonderful lectures, great clarity for complex things. Same for myself, of course, and same for Shona. And Ricky, I think you've done a valiant job in the background, just making sure everything works with all this complicated toing and froing in and out of the different rooms.
Thank you.
Go ahead. Oh, you've written on the chat, yeah.
Yes, please fill in a survey, give us all your feedback. I've attached it here to the chat, but I also will be sending you guys an email with it as well.
Yeah, that would be really helpful to us, because obviously we've never run this virtually before, and we would quite like to get your sense of what worked and what didn't work. So if we run it again, we can modify it in ways that make it even better. But,
and again,
okay.
feel free to reach out and ask us questions. And if we write you back, then that means we're interested in answering them.
Right, I say goodbye. Thank you for attending.
Thank
you. Good night.
Thank you to everybody. Good night. Good morning.
Goodbye. Goodbye. Thank you
very much.
Thank you.
Thank you. It was great.
Thank you.
Thank you all.
Thank you very much.
Thank you.